As always. So how did week one go? It looks like we're still missing quite a few people. So, has anyone heard of any problems with anything of why people aren't coming or uh yeah, don't worry. Well, you can right click on your name, I think, and change it, but that's okay. I got you. Um, Jennifer from your last name. I was able to find out that it was you. All right, so this week we get to talk about different types of media, right? So let me go ahead and share my screen, and we are going to work through a couple of fun little practice questions. And like I said, make sure, I mean, as for right now, you can still use your book and everything because I know that this is all very new, but make sure that you do study up on this as well. Alrighty, so for the first question, what do you guys think about this? A nutrient media that has all of its chemical components identified and the precise concentrations known and reproducible would be named what? So this one's just going to be a definition. When you guys come up with the answer, you can come off of mute and say it out loud. Are me recording the videos and putting them up working okay for you guys? Yeah, okay. Does everyone have their book and everything by now? I know that there was some problem. Okay, there were some problems with the bookshop getting on time and stuff. So I still don't have mine, but I was able to get electronic access to it. So
I think I know the answer, but it might be wrong. I couldn't
that's
find
okay.
it in my book. Is it A?
Yes, it is
Okay.
absolutely A. It is synthetic. So great job. So yeah, synthetic is going to be, like I said, and this was just kind of a definition, so I don't have any fun ones, but I would think of it like synthetic. We synthesized it, we made it, so we know exactly what goes into it, right? Because we are like figuring out what we want to put in there. Right, awesome, very good. So let me, I guess I probably shouldn't highlight my screen with a highlighter, right? That would be something I would totally do. Use a highlighter on my computer. All right, so question two, bacteria that require special growth factors in complex organic substances are called what? What type of bacteria? And again, your book does have some great charts on this that summarizes everything.
Is it A?
Yes, fastidious. So when I kind of think of, I don't know this word, like I said, you guys have probably learned by now that I really teach outside of the box, but if you're being fastidious, you're being very, very picky, right? And so bacteria that are being picky and they're requiring special growth factors and very complex organic compounds or substances, they're being very fastidious, so very picky. Anaerobic is just without oxygen. Aerobic is with oxygen. Pathogenic, that's for one specific pathogen that you would, so it'd be like more of a selective. All right, the 6 I's, your book really likes to stress these, so make sure that you do know these 6 I's by memory.
Is it C, infection?
Yes, good job. So, and this would just be, again, a definition flashcard of what those 6 I's are. And so the one that is not, because it says except, would be infection. Yeah, because we don't want to infect our cultures. You know, we want to inoculate it on purpose. But an infection would be like an uncontrolled one, where if you like, you know, it always amazed me because I used to pour plates when I taught this class down in Kentucky because I worked at Campbellsville University and I taught med micro for when I was down there. And you know, you'd pour your agar plates and you'd like leave the lid like slightly tilted off so that it would like not get a ton of condensation. I'm like, how is it not getting infected? But I never did. But anyway, yes, so the infection is going to be one that you don't want, okay, because that would be contamination and then you wouldn't have a pure culture. All right, what about little Miss Sally? Again, I'm all for root words and picking a word that kind of triggers something.
Is it A?
Nope, but good
No.
one.
Okay.
So this one, like, it's prior to arrival. So this means that it's gonna still have to be transported, right? Because it's being taken at the office and then it's being transported to the clinical lab. So this one, that's why I said kind of look for any root words that would work. Transport media, because like TSA plates, these are plates that you know put in the incubator to grow for 1 to 2 days. Selective differential agar, those are going to be growing plates. And TSB tubes, those are just going to be like a nutrient broth tube. So the best one is basically just transport media, because you're not really encouraging selective growth or growth really at all. It's just kind of a maintenance before you can get it to the lab and set it up. But I love the guess, so thank you. Oh, oops, I didn't mean to. Yeah, that's okay. All right, so the answers explained in the paragraph below. So how will E. coli appear if the mordant is not applied?
Is it D?
Close, but it's actually going to appear as gram negative, because you're still staining the first part of it, but not the second part, right? So you're gonna get some color, but not the fixative. So it'll appear as if it was gram negative then. So the mordant here is gonna be like the fixative or the second color, right? So the iodine is added as the mordant to form the crescel violet iodine complex, that blue color, so that the dye cannot be removed easily. So if we didn't have that, then it would just appear as a gram negative. Right. I think it's crazy that they really have agar named chocolate agar that has chocolate in it, blood agar that has sheep's blood in it. Crazy. Like I said, there's a nice table, and I even put it in here as table 3.8. So open up your book and look at 3.8.
Is it gonna be C, Sin?
Yes, it is sim.
Okay.
So by looking at that table, right, we can figure out we're looking for just the mobility. Okay, so like chocolate agar is going to be specific for, nutrient, etc. So the one that we would want to look for would be the sim media, and that will help us look for mobility. So very good. Okay, so a microbiologist takes a smear of bacterial cells, stains them with loafers methylene blue. That's what I dropped on the floor when I first started in Kentucky. It was great. All of the cells appear blue under the oil lens. Oh, I spelled lens wrong. This is an example of what? Can you tell I teach anatomy? So I would look at a table in your book that talks about different types of staining techniques and why you'd want to use them. But methylene blue is a pretty simple stain.
Is it simple staining?
It is. Good job. Did you use a table for that, or was that a good guess?
I used the table. I can't
Awesome.
lie.
I'm glad you did. What page was the table on?
I think it's on 72.
I can't wait until I get my actual book instead of the online version. But yes, good job. All right. So what method often results in colonies developing throughout the agar and then some colonies on the surface? So keywords here: throughout the agar as well as some on the surface.
Is it gonna be C, a spread plate?
Close, but that would actually just be on top. Same with the streak, that would be the top. where a pour plate, you're actually mixing the
Okay.
bacteria into it before you pour it. And so because you're pouring it, it's going to be throughout the agar. Where a streak plate, you would just, you know, you'd take your plate and you'd streak it back and forth really carefully. And then you might do a turn, like in a sterilized turn, streak, sterilized turn, streak. But that'd be a streak plate for like cereal dilutions. Whereas spread plate is you take your sample and you put it in the middle and you take these glass hockey sticks. They're actually kind of cool. They look like an L. You sterilize it first and then you just kind of spread it all over the top with the hockey stick. And so that's still just on the top. Where when you're doing the pour plate, you're inoculating the media before you actually pour it into the plate. So it gets throughout the layers of it as well as on the surface. Love that you guessed, so thank you very much. So it'd be a pour plate. All right. Common media used for growing fastidious bacteria is, and again, use those tables. Tell me what page you see the table on when you get to it. Rebecca's dog's so cute. Oh no, that was a cat, long tail. Looks like a little pug for a second. Wait, did anyone find the page with the table on it yet?
Is it chalk B?
Yes, it is chocolate agar. What page do you find the table on?
Well, I found the information on 81
Okay.
by the definition of fastidious.
Awesome. So yes, this would be a chocolate. I think it would be fun growing chocolate. Like Willy Wonka and the Chocolate Factory, right? Jenna, you're so funny. All right. So the next question, a student was given a stain smear to observe. Upon observation, the student noted that rod-shaped bacteria were clear and unstained against a darker background. So what type of stain was performed?
Is it D?
Yeah, negative stain. Good. So how did you know that one? Or was that just previous knowledge, or did you find a table?
Previous knowledge.
Awesome. Yep, it would be a negative stain. Kind of with the contrast is what I think about that. It's like looking at a film negative. You guys might be too young to remember film negatives. Back in the day before digital cameras, we used to use film, and the film negative was dark against light. And so that's how I kind of remember them. And then also the shape of it, it's specific for, well, it's used for like rod-shaped bacteria. So good, negative stain. Awesome. All right, now I know that we don't talk a lot about microscopy, like they had you guys skip the microscopy part of this chapter, chapter 3, but I know in lab today we are looking at some microscopic techniques in my lab. So some of you guys aren't in my lab section, but in lab this week. And so, and since we do talk about and use a lot of microscopes in this, I thought that this was still an important question. So why is immersion oil used when viewing specimens under a microscope?
Is it C?
Yes, it is to increase the resolution. Good job. So if we think about it, you know, it's kind of like if we look through my water bottle, things are like distorted, right? But if we fill this with oil and we look through it, it's going to be a crisp and clear image. So you only use oil immersion on specific objective lenses. I mean, don't use it on a non-oil lens or else you'll ruin it, or somebody will have to spend a lot of time cleaning it. But yeah, so the oil immersion is just going to be for your higher magnifications, and it gives you a much clearer resolution. So like a lot of the 100x objective lenses are going to be oil immersion lenses, and then don't forget to add the ocular eyepiece of 10, so 100x plus 10 is 1,000 times magnification. But yeah, so your oil immersion, that's my mouse works for that computer, Janet, not this computer. Okay, so that one would definitely be to increase resolution. So very good. All righty. The term that refers to the purposeful addition of microorganisms into a laboratory nutrient medium is called what?
Is it inoculation? A.
Yeah, exactly. So if you think about like, and I always stress this in every single class that I teach, and you know, always try to take things outside of the question. Okay, inoculating, you know, we'll talk about inoculating loops and, you know, different ways to inoculate or purposefully put in a microorganism. But if you think of contamination, okay, if you're ever making, like I used to make cheese, and, you know, a mold would be a contaminant. Okay, so contamination, you guys are going into the medical field, you don't want contamination because that's going to just lead to problems. So that's the non-specific organism that would be growing in whatever you're looking for. Isolation, this would be like doing cereal streaks or you know, colony picking to isolate, or like solitary, I love watching prison shows, so solitary confinement is isolation, right? So isolation is just taking out one colony to pick on. Infection, well, that's, you know, getting sick or infected, or, you know, more than your regular microbes. Immunization, these are shots against something, right, to bump up your immune system. And so when we want a purposeful addition, that would be the inoculation. A lot of these others are non-purposeful. So contamination is not purposeful. Infection is not purposeful. Good. Tomato juice louder. Okay, which of the following nutrient media is used for cultivating fungi? And again, when you find the page number, let me know and I'll write it.
It's on page 82 in the table 3.7, and I believe it's D.
Yeah, try to pronounce it. Gonna try it.
Sabaroda?
Sounds
Saba
good.
something?
Sabaro. Sabera. It's a pizza place in the mall, right? Okay, yeah, Sabaro. Like, I butcher people's names, and, you know, it's so funny. I'm hyphenated. My name is Miller Monfils, and how many people like butcher Monfils cracks me up. So anyway, but yes, so I will never hold you true to pronunciation. So, okay. All right, so we'll do blue. Very good. Awesome. So this is on page 82. Table 3.7. All right, cool. And which of the following reagents reacts with cresol violet as the mordant?
Is it C?
Yeah, it is Graham's iodine. Good job. So cresol violet is going to be, it can't react with itself. Well, actually, that would be the same. But the Graham's iodine would be that chromastin. So good job.
I have a
Alright.
quick question.
Yeah, what's up?
Can these questions be put in our in your class module thing or anything? Like
Sure,
where you
I
put
mean,
your videos?
it can be, or actually this lecture is recorded, and so
Oh,
then
okay.
I can, I'll put the link to the recording up, so then you can rewatch my smiling and frizzy hair.
Okay.
Yeah. So do you guys
I
like
just wanted
these
to write all
questions?
these questions down. Yeah.
Yeah, you guys like these? Okay.
Yes.
Cool. Yeah, I went to the, so like I said, I got access, well, I'll tell you about this in a minute. Okay, so yes, so good job. Okay, what about this guy? This is kind of relevant in the middle of making a COVID-19 vaccination too. We'll talk about that actually.
Would it be C?
Yes, it is. Live media. Okay, so the thing with this one is we're using a chicken. Okay, a chicken is still alive. And so that's how you can kind of remember that it's going to be live media. Selective media, remember, just like by definitions, right? Selective is going to select for one thing. You know, complex media is going to grow multiple, microorganisms possibly, but in one category. Enriched media is specifically for, whatever that is, something that is iodine loving or whatever. Where live media, to me, live is, live. So, yeah, so that was a very good one. All right, so that is all the questions I have. So Janet, mental note, we need to put more questions in here. Books are supposed to be, okay, so let me catch up on chat. I don't have 9 books. Books are supposed to be here tomorrow, Monday at the latest. All right, yeah, so if you guys don't have books, don't freak out. Like I said, I still don't have a book either. And like I'm giving you the lecture recordings and the lecture slides. No, I haven't uploaded slides yet because I just found them. So I will work today or each week to put the slides up that I make the videos on so that you guys can print them off. The reason I say that I'm making the videos on is because, like, for instance, this week we only talked about 3.1, 3.4, and 3.5. So I wouldn't want to put all of chapter 3 slides up there for you without editing them first, because I don't want you guys to have to study stuff that you're not going to be tested on, right? So as I get ready to make the lectures each week, I'll post those videos. So I'm going to make myself notes. So when I post videos, I will post the slides used, and then if you want, I mean, I can post these ones too, or you can just rewatch the video. What would you prefer? You just
I don't mind watching the video.
So then that works. I'm gonna leave it like that because then because I'll probably teach micro again. And if I put all the slides out there, I know that things get shared, and that's completely fine. Like, I have no problem with that. But then, people could come to class knowing all of the answers already and not try. And so I'd rather not post these slides, but if you guys want them, I totally will. All right, so notes for myself. I will, when I post the videos, I'll post the slides that I use to make the videos, and then I'll post the lecture after it's done uploading and doing its buffering and stuff like that. And then I'll have to find more questions for the next time, because this only took us a half hour. You guys are super smart. But yeah, but what I was going to talk about is, so a little bit of current topics, right? With the COVID and Corona going on and everything still, what do you guys think about this vaccination that they're trying to push through? Do you guys feel confident in having a vaccination before it's like really tried out? Or I don't know, like, let's discuss this. What do you guys think?
Yeah, I guess, I don't know. I've read way too many things about it, and some people are saying like they're trying to put chips in us with the vaccine and all that crazy stuff. And then it's like the people that don't feel comfortable with vaccinating their kids, but the parents themselves are vaccinated as kids. So I don't know. I guess people do have their differences when it comes to vaccines and the effects that it could have on their kids when they get older.
Uh-huh.
But yeah.
Well, I mean, I actually know some very smart people that are anti-vaxxers. So, I think that is a personal choice. I mean, me personally, I'm a pro-vaxxer, but I'm not going to push my opinions on anyone. So, I mean, we can have a fun little discussion about that right now as I'm updating the attendance list. But yeah, I don't know. I mean, I like that there is a vaccination that's like happening, but I think pushing it through without all the proper clinical trials is a little fast. And like me personally, I won't get it right now because there's too many other underlying issues with my own personal body that I don't want to mess it up. But I don't know, what do you guys think about that?
Yeah, I would. Go ahead.
I just think it's kind of scary because we still don't really know all that we can about Corona, and they don't have answers to everything. So how are we about to sit there and say that everyone can get this vaccine when we don't really fully understand the disease itself yet?
Yeah,
that's what's scary to me.
Yeah, the problem is that, things are gonna, like science, it takes so long to actually remember, or to not remember things, to learn things. And so that's where like a lot of these conspiracy theories come in and stuff like that right now, is because, they're like, oh my God, well, you keep changing information, so you obviously don't know what's going and it's being made up. Well, no, that's not the problem. It's just that science like evolves all the time, right? And so we really don't have a legit answer, and we keep learning all the time. So yeah. All right, what are other people's opinions on it?
I heard that they wouldn't have a vaccine by the time that election was here. I didn't really read anything else on like when it was actually coming out, but I heard that it won't be here in time for the election, and everyone's saying that it's just a big blow up thing just to get Trump out of the office. And
Yeah, well, and like a lot of people try to make everything politics related, right? And some things just aren't. And so that's where I think that, a lot of people are trying to say, oh, love or hate the man, he is our president right now, but they're like, oh, Trump made this up. No, it's a legit thing. I mean, I have doctor friends that are in the medical field losing people to COVID every day. It's not a made up thing. But, people try to politicize everything. And, just like you said, the vaccination is not going to be ready by election. Good. It shouldn't be. It's been around less than a year. I don't trust it. And again, I don't think that this is politically driven at all. But I mean, again, those are my personal opinions. But I mean, anyone that would stoop so low to, you know, release a virus that's killing hundreds of thousands of people would be, you know, they should roast on a popsicle stick down with the devil, you know, my personal opinion. But yeah, I don't know. But I mean, it'll be interesting because, you know, since we're talking about micro, you know, we're talking about how vaccinations are made and stuff, you know, this is why I love science, because, you know, we can actually pay attention to what's being said and then be like, yeah, no, that's just your opinion, or, you know, that's fact. Like, obviously, we know an article coming from theonion.com is not going to be legit, right? But other places should be giving you unbiased opinions, but, you know, sometimes they don't do that either. But you guys are all smart enough to be able to use your brains and be like, yeah, okay, no, that's just totally your opinion. That's not based on science at all, you know? So that's why I love science, because, you know, we get to learn how to, you know, figure out our brain, right? To be able to say, yeah, okay, I'm going to use my brain, and say, you're totally making stuff up, you know? All right, let me try to figure out who else is here. Okay, I'm looking for Athena Adams. Athena, anyone? Autumn Amorino? Selena Bibb? Callie Briggs? Natalie B? Deborah, I saw you in here earlier. I have you checked up, of course. Summer Jackson? Rachel McPherson? Julia Parker? Riley Richardson? Haley Swan Chubb? Alexis Ward? Anya Whiting? Or Malia Zidapeko? All right, so yeah, I mean, like I said, you know, we're in such a really interesting time right now in the world, because, you know, all these things are happening and going on, but it's also very scary that all these things are happening and going on. You know, there's the fun little meme on Facebook that I found last night, but it's been going around forever, you know, about kids trying to learn about 2020 in history class in a century from now, and like they'll just break a highlighter over the whole page and like spread it around, because there's so many things that happened this year and everything. But, you know, again, it was kind of, I mean, not fun, because nothing about COVID is fun, but I was teaching pathophys when COVID was launching, like it's a thing that we chose when it first came about in, like, you know, January. And so we followed the progression of it throughout the whole semester. And, you know, now since we're talking about it in micro, you know, we're going to talk about how vaccinations are made and really cool. All right, are there any questions about anything? How many of you guys already had lab this week? Probably about 3 or 4 of you. Hopefully it went well. Yeah, Rebecca, did it go well? Chris taught it,
I
so.
actually got added to class late, so I missed the first one of it. So the second class, I was really lost. But I have Professor Woodley, and he's super nice, so he's gonna help me like make sure I get caught up. And he said no worries until I get my books and everything.
Exactly.
So.
Yeah, Chris is awesome. And, you know, all of us, seriously, like, you guys are so lucky to have, I know Chris really well, and then me, of course, but you know, Baker people really care, and so sometimes they get really kind of stuck on dates and stuff. But like I said on the very first day, I'm all about life happens. I don't want you guys freaking out and saying, oh my god, I'm 2 weeks behind, I just should drop out because I'm not going to catch up. No, dude, we have 16 weeks in this class. If you're 2 weeks behind, we'll have to work a little bit harder to get it to work, but there's time to make up for it, so don't freak out. All right, so yeah, so for next class, I'll probably aim to have more like 30 questions, because you guys are cranking through them nice and fast. But I kind of like this format. I would like to get any opinions from you guys, but I mean, recording the lectures so you can, you know, look at them on your own after the fact, I think is fun, because then you can revisit them. And then these are recorded so that I can put them up for you too. but going through questions more like the application based. So you guys can listen to me on your own and then just come to class and we can do questions together. Does that work for everyone? Yeah. Cool. Cool. All right. Are there any other housekeeping things that we need to take care of before I let you guys go?
It looks like you have a question in the chat. Somebody said that the book's lab is different from the lab instructions on Canvas, and they want to know what lab we're doing.
Thank you. The lab is supposed to be in module 2. So let me, you guys can still see my screen, right? Oops.
No, we can't see your screen. We can only see you.
My beautiful face. Oh, right. I'm going camping today. I'm so excited. Share screen. Share. Share that. All right, how about that? All right, so we are in microbiology lab, and then you can go to modules here, or it pops up like that too, and then just scroll down to we're in module 2, and this is your reading assignment, and then your module 2 lab exercise 3.1. So when you click on that, then it'll bring up module 2 lab directions, and then the proper care of the microscope and slide drawings. So then you can open up these guys and print them off and everything. So
If we can't print them off today, is that going to be a problem?
no, bring I'll print off a few for today, but you know, just try to do it for next week, you know, because
Okay.
but yeah, I mean, I know that yeah, I'll just print off a few today. So Alexis, does that make an Alexandria? I thought I emailed back to you, Alexandria, or made a class announcement pertinent to that, but what questions did, and Alex and Alexandria, what other questions can I help you out with?
Well, okay, sorry, I'm gonna be kind of quiet because my roommates are sleeping. But those directions and the directions from the lab, those directions and then the directions from the lab book are completely different. They're two different experiences. Like if you click the lab exercise and it tells you to read read the experiment in the book, and those experiment directions are completely different.
Then I would follow the experiment directions that are written out on Blackboard. But I think like I think we ran into this last week too, where there was a couple of things that were written slightly different, but it still gets the same point across. So, well, like I said, I don't have a lab book, so I'll have to work with you, Alexis, in class. But I would say your go-to is going to be what's posted on Blackboard.
Okay, thank you.
Yep. All right, Alexandria, do you have a question, or did I answer your question, or do you want me to stay after and you can talk to me? Alexandria?
So you did receive my email then?
Can you say that again? You went out.
You did receive my email then?
If I didn't address it, then I thought I probably did. So can you either stay after class when I stop recording and ask me, or if it's not a personal thing, you can ask the whole class right now, whatever you would prefer.
I can stay after.
Okay, cool. All right, so are there any other questions right now? No? All right, so pack up, and then we will see you guys in lab, or those that already had lab. Have a fantastic week, and we will see you next week. All right, I'm gonna stop recording.